Okay, let's talk about inhibitory synapses. What I have drawn here is a nerve cell. Okay, so we've got a nerve cell there, and it's got three synapses shown on that. There's two blue ones. There's these acetylcholine ones. Okay, so these are incoming nerves which are going to release acetylcholine as a neurotransmitter, and that's going to affect the membrane. It's going to Well, acetylcholine, remember, it binds to a sodium channel and lets a little bit of sodium into the cell. And the same is going to happen to this acetylcholine here. These would be called stimulatory synapses. This one here we haven't come across before is this nerve doesn't release acetylcholine as a neurotransmitter. It releases this substance GABA, is gamma aminobutyric acid. Okay, now that works on a different ion channel and has a different effect on the cell. Let's just think about the acetylcholines for a minute, though. We go over to our action potential graph here. Right. So we've got the resting potential there of minus 70 millivolts. And I've drawn in the threshold potential there of minus 55. So if I just draw a dotted line there. Okay, so imagine one of those acetylcholine receptor fires. It's going to let a little bit of sodium into the cell, right? But it's not enough to reach the action potential, the threshold potential, sorry. So no action potential occurs. It just goes back to the resting value. Now let's imagine if both of those fired, so you'd get a little bit more sodium coming in. So it would go up a bit for one and the other one fires, and it would just take it over the threshold potential. And yes, you would get all the voltage gated channels would open, sodium channels, and you get an action potential. Right. So that's how those two synapses work together. And let's see what the GABA one does. Right. So let's look at this little chart here. Now, when acetylcholine binds to the postsynaptic membrane, the ion channel it opens is sodiums. So what will that do to the potential inside the cell? Well, it's going to release positive ions. It's going to become less negative. It's going to go up. GABA, now that doesn't act on sodium channels. That it binds to a receptor for GABA, and that is a ligand-gated channel. That opens up chloride ions. Now inside the neuron, you've got a low concentration of chloride ions. It's higher outside. So when you open up those channels, chloride will flood into the cell and it will lower, because chloride ions are negative, it will lower the potential. So if you draw a little graph here, let's see what happens if a GABA receptor fires here. Well, it's going to do that. It's going to make the membrane potential lower. So that means now, if both of those acetylcholines stimulate, so let's say that causes an influx of sodium up to there. Now, under normal circumstances, if you start off at minus 70, that would take you above the threshold value of minus 55. But because we're starting from a lower base, because of the action of the GABA, then it doesn't. So the nerve doesn't fire. So that is how inhibitory synapses work. Then they're usually through this neurotransmitter GABA. That will lower the membrane potential, making it harder to trigger an action potential. Now, a lot of the job of the nervous system is apparently just damping things down and stopping nerves becoming too active and firing too often. And obviously, when things go wrong with the nervous system, very often one of the things that happens is you get seizures and fits, and that's because there's too much activity. So GABA is really important in lowering the action potential and damping down the nervous response where needed. Okay, so we think about let's think about some well-known drugs. So we think about benzodiazepines. So These are things like Valium, Xanax, sleeping pills like Temazepam. These are all what these do is they bind to the GABA receptor. They don't actually they're not GABA agonists. That means themselves they don't mimic GABA itself. Bind to the GABA receptor, sorry. But what they do is they bind to a part of the receptor. That's not enough to trigger the receptor, but they bind to it and they make it easier for GABA to then trigger, to bind and to trigger it. So all of these kind of drugs make people relax, and they damp down the level of neuron activity. Another one of these would be drugs like phenobarbital, phenobarbital. And that is an anti-seizure drug. It's used in to control epilepsy, and it stimulates. Well, it binds to the GABA receptor, makes it easy to be triggered, damps down the activity of the nervous system. Okay, so it's the balance between the stimulatory receptors, the stimulatory synapses, the acetylcholine, and these inhibitory ones, GABA, which determine how easy or how difficult it is for a nerve to fire.